webináři, který se bude týkat produktu Reaxis Medicinal Chemistry. Dovolte mi, abych přivítal Jamese Rinkera, který vás dnešním webinářem provede. James pracoval přes 20 let v oblasti medicinální chemie ve farmaceutickém průmyslu, ve vývoji léčiv a v současné době je zákaznický konzultant a produktový expert na databáze Reaxis Medicinal Chemistry a další produkty z oblasti Life Sciences, zejména Pathway Studio, což je produkt, který doporučuji, abyste si také eventuálně prohlédli. Já bych mu předal slovo. Dear James, it's your turn now.
Okay, so I'm going to go through some slides right now. I just want to give you a little bit of background on Reaxis medicinal chemistry, how it's built, what kind of information's in there, the amount of information, and then I'm just going to go to the live demo portion. I'm going to do a few examples just to give you a flavor of what it can do, and then I'm just going to open it up after that for questions. And maybe if somebody wants to do a search, we can try to do that search live. So just as a intro, Elsevier has a suite of life science tools that we're working on integrating into a solution for research and development, and the tools that we're actively working on for R&D are Pathway Studio and Reaxis Medicinal Chemistry. Reaxis is more of a purely chemistry tool, while Reaxis Medicinal Chemistry sits on top of Reaxis, so you would have all the capabilities of being able to do reaction substructure searches, finding physical and spectral data, but then we add on top of that all the biological data with Reaxis Medicinal Chemistry. And then Pathway Studio is integrated with Reaxis Medicinal Chemistry, where you would look for disease targets and diseases and cell processes and relate all those together and understand those processes, and then bring in the target information into Reaxis Medicinal Chemistry to find actual data on those targets. So what's in a relevant medicinal chemistry paper? Chemical structures, druggable proteins, bioassay data, quantitative results. So what we do is we take that journal article and we pull out all of the citations, all of the druggable proteins, chemical structures, quantitative results, and bioassay data, any tox data, PK data, etc. All this is extracted out of journal articles and patents, and it's put into Reaxis Medicinal Chemistry. And the biggest difference between this tool and other tools, like if you thought of a tool like Chemble that's online, is the high rate of curation and the amount of data in it. So the data is curated to a high extent where you can search for chemical structures, or you can just search for targets and find all chemical structures, but you can search for them in a way where you can combine several different fields and only find the hit sets that you are actually looking for. So, and it covers the patent data, which is actually probably 90% of all the biological, the bioactivity data is in the patents. So if we just look at the coverage, there's about 25,000 patents, world patents, that are covered from 1978 to present, and another 22,000 U.S. patents that are covered from 1971 to present, and about 5,000 European patents covered from 1979 to present. And this isn't a lack of European patents. This just means that there weren't as many European patents as there were world and U.S. patents. So 50,000 total patents that are covered. And there's over 1,000 journals that are covered by Reaxis Medicinal Chemistry from 1980 to present. And you can see the top 40 journals here. These should be very familiar to anyone working in the medicinal chemistry area. JMedChem, BioOrgan, MemChem Letters, BioOrg, MedChem, European Journal of Pharmacology, etc. And there's over 200,000 articles that are included in Reaxis Medicinal Chemistry. So all of these journals are actively being read. All the relevant data is being extracted out, and then it's being put into Reaxis Medicinal Chemistry. So basically the bottom line is the essential information that you would need as a medicinal chemist to understand structure, activity, relationships between a compound and their targets, or a compound and cell-based assay. So understanding if a compound is being transported into a cell to get to an intracellular receptor, animal disease models. So any model, you could search on specific animal models as well. So if you had like a mouse model that was for rheumatoid arthritis, let's say, or glaucoma, any kind of animal model, you could search for that specific animal model as well. And all the PK data, as long as the PK data is reported for that compound, whether it be oral bioavailability or clearance or volume distribution, all that data is actively extracted and put into RMC. And then finally, all the tox data as well. So tox data can be either reported as a lethal dose, or it could be reported as off-target activity or herd data. And that's it for the slides. I wanted to make that part very brief. And I'm just going to go into RMC now, and I'm just going to give you an overview of some of the basic features. I'm going to give you a little bit of a tour, and then we're going to show how this can be used in a workflow, and then we'll do one or two examples after that. So there's many different fields that you can search from, and you can do a substructure search, and everything is related to the structure. So we can do a structural search related to either a target or a bioassay category. So the categories are broadly classified as in vitro efficacy. So this would be all your target-based efficacy, whether it be a kinase or a GPCR or whatever. And then all your in vivo animal models, and then metabolic data. So all your cytochrome P450 activity would be in the metabolic and transport data. Also all your KCO data, blood-brain barrier permeability, and then PK data is broadly classified as well. So you can either select on, say, PK data and look for all PK data, or you can, in the parameters field, which I'll show you in a couple minutes, you can put in specific parameters for either clearance or volume of distribution and search in that manner as well. And then all the toxin safety data. If you go under target name, all the targets are actually classified according to protein classification. So you can go under nuclear hormone receptors, and all of them are classified either as the estrogen receptor or the thyroid hormone receptor, etc. And then you can tree down underneath that, and you can just get down to families of receptors. And from those families of receptors, you can get down to the individual ones. And here, the other thing that I wanted to point out is that when you get down to the individual receptor, you can limit it by species. So here, bile acid receptor is either rat, human, mouse, or hamster. So if you only want to see human data, let's say, or you only want to see mouse data to do comparative analysis, you can click on the human-only data, and then you can do direct comparative analysis, human-to-human, for a specific receptor. So I could select one receptor, bile acid receptor, and I can compare that to something in the estrogen family. And then I can transfer that over, and I can look for that data in RMC. So parameters. So sometimes there's two ways to search for bioactivity. One way is with something called a px value, and what the px value is it's a logarithmic measurement of activity. So for reported activity, some compounds may be reported as having an IC50. Others may be reported as having a % inhibition. Some may be reported as having a KI. So what the px value does is it normalizes all the data and gives you a relative activity for, across many different parameters. And so with the px value, I can do comparative analysis across many different targets. And even though I may have an IC50 for one compound and I have a % inhibition, I can get a relative idea of how active that compound is. And then I can go to the actual data in the substance report and find out what the actual data is. And then unit is just the unit of measurement, either nanomolar or micromolar, and then the actual quantitative result. Under parameters, we can look for things like IC50, or we can look for things like clearance, which is the actual clearance for PK data, or we can look for PAP, transport, which is a measurement of permeability in cells like Keiko cell lines. So this would be used to either find, to either do comparative analysis on Keiko data for a measurement of oral bioavailability, or it can be used with MDR cells as a measure of blood-brain barrier permeability. So depending on what you want to do, you can use the different fields to actually find the data that you're looking for. And then you can add or remove fields as well. So on this screen, I only have some of the more relevant bioactivity data. You can go into the bioactivity data and it just tree down under medicinal chemistry, and you can add or remove fields. So you may be looking for species, should be one of them under here. Oh, I already have it on there. So under biological species, you can just limit all your data to either rat or human or mouse or whatever species you're looking for. There's actual mutant or chimera data for the targets. So, and I'll show you that in a moment when I do a comparative analysis. Not only is it going to show the broad heading of the protein, but it's going to also show any of the variants of the protein as well. But you can limit it by that variant up front, or it'll show you all the variants. So, all right, let me do an example. So I want to show you how this can work between two different products. So what I did is I started to build a model for psoriasis, and in this model, in the disease of psoriasis, T17 cells are activated in an autoimmune way by a number of different cytokines, either by TNF or by IL-6 or by IL-23, which is IL-12B. It's another synonym for it. And then I began to build in all of the known drugs or drugs that are in clinical trials against psoriasis. And here you can see some familiar ones: Humira, Stelara, Enbrel. And then what these do is they go through different pathways. to activate IL-17A, which is a key cytokine regulator in inflammation. And so depending on what pathway these go through, you can envision some of these kinases as being possible small molecule drug targets for either psoriasis or rheumatoid arthritis. And so some of these that have been implicated in the pathway have been the JAK kinase inhibitors here, or the JAK kinases in this pathway, which activates STAT3, and then STAT3 is responsible for binding to the binding domain, which is responsible for transcribing IL-17A. So STAT3 is not a very good target because it's broadly expressed in all proteins, I mean in all cell lines, and it's broadly expressed across the human body. But JAK is a more druggable target, and it's only expressed in either this disease state or in cancer. So we're going to take a look at JAK 1, 2, and 3, and then we're going to see if we can get some selectivity over JAK versus other kinases, because there's a ton of different kinases that you wouldn't want to hit. So I'm going to take the information from this pathway here, and I'm going to move it over to Reaxis medicinal chemistry, and we're going to see what we can do with that data. So one of the ways that you want to approach this problem is you want to search for JAK individually, and I'm going to look for all the more active compounds, and I'm putting in an arbitrary number, but I know from my experience with Reaxis medicinal chemistry, the px value of 7.5 is approximately 10 nanomolar or below. So I'm going to do the search for all JAK1 compounds that are greater than 7.5. And I'm not going to do JAK2 and JAK3 live. I'm just going to show you how the problem is approached. And so if I only searched for JAK1, it's only going to bring back all JAK1 data. It's not going to bring back all the other data. Then what we would do is we're going to take that data and we're going to go up into the history. And the history is very nice because what the history allows you to do is to save your searches. And then from the save searches, and I'm going to show you the save searches. So I saved these searches down here before I did the webinar. And then we can combine all of these, any of these that we want together. So if I'm looking to compare all the JAK compounds together, and I only want to find a list of compounds that are active against those three kinases, I would do the search 3 different times. So I would search for JAK1, JAK2, and JAK3, and I would search for all those greater than 7.5. If I search for those 3 all at once, it would search for either JAK1 or JAK2 or JAK3. And here I want to combine those. I only want to find those compounds that are active against all 3 of them. So once you do those searches, you can come into the history, and you can select those 3, and then we can go up into the history, and we can combine those hit sets together, and we would want to find the overlap between those 3. And so this is going to give us a list of 875 compounds, and I'm going to go into the heat map and show you what the heat map looks like. Now, unfortunately, we just came out with a new version of RMC, and they're working on fixing a bug that's in it, because this should only show the JAK 1, 2, and 3 data, but it's showing all data. They're aware of the bug. This thumbnail, it allows you to scroll across the data, because there's a lot a lot of activity data here. So it allows you to search across this entire scheme and find where most of the activity is. So here we can look at JAK1, JAK2, and JAK3 inhibitors. So all of our data, we know that for all this data, these are all highly active inhibitors that inhibit all three of the JAK kinases. And then we can compare that to all of the other targets that those hit or don't hit. And so what we're looking for is selectivity against those targets. So here you can see for some compounds here, this is a staurosporin analog. It looks like it's non-selective. It's broadly active against not only the kinases that we want, but also other kinases. And if you look at this compound here, it looks like this compound is actually very selective. So we can scroll across and we can look at its activity over a number of different kinases. Also receptors here, you can see e.g. RF is FR is here as well. If we want to limit this to just a battery of kinases that we would be interested in, we can do that as well. So I'm going to go over here and I'm going to go back to the query again. And I saved the reaccess IDs. We're working on creating a seamless transport between the history and transporting them into the next query. So you would just want to take all those compounds that were active against JAK1, JAK2, and JAK3 and then go into the target name and just look for a battery of different kinases that you were actually interested in doing the comparative analysis on. So we can go under the classification of enzyme and look for transferases. And under transferases, we can go under kinases, and then all the kinases are grouped. So I can go under the serine threonine kinases and look for atypical kinases or ATP kinases. And so I can do a comparative analysis, let's say, of all the AKT, DMPK, PKC, B, A, and then I can go under the tyrosine kinases where JAK resides. And I can look at JAK and, let's say, ABL and SARC. And I can transfer all these in. So they're classified in a very meaningful way for you to be able to do comparative analysis. And then we can search for all those bioactivities. So here now, instead of having a huge heat map with all of the data on it, now I have a heat map where I can do a direct comparative analysis between only the inhibitors that I'm interested in, plus a finite battery of kinases that I want to know if it's hitting them or not. And so here you can see this compound is obviously very clean against all these different targets, but it's highly active against JAK. And the one thing I want to point out is with these numbers, so the difference between, say, 5.5 and 6.3 is one logarithmic scale, so that's actually tenfold less active. So 5.5 is tenfold less active than 6.3, and these are actually 1,000 times less active than the activity against JAK. So these are actually very, very, very selective. I could then go and look at this data, the real data, not the comparative data. So we can go and we can say show details for that particular compound. And so this was actually one of the compounds which was on my pathway to Factinib, and this is a compound that I think is actually on the market now for rheumatoid arthritis. So we can do the comparative analysis with, and then we could see TYK2 is actually in the pathway as well. So we can go under bioactivity, and you can see all the data here, not just the in vitro efficacy, but you can see the metabolic data is here. So all of the comparative analysis between its activity against the cytochrome P450 battery, and some other assays here. There's some clearance data under PK, all the Cmaxes, your volume of distribution, your Tmax, all that data is here. We can go out to the actual paper. We can view the title and abstract for that paper, and we can go get the full text article if we have access to the full text article. So, and I think this is an Elsevier. No, it's not. It's the Wiley Journal, but if your institution has access to it, it'll take you right to the vendor's website, and then you can just get the PDF and download it. For the data that we were looking for, we can go to in vitro efficacy, and then it'll take us to all of the in vitro efficacy. So here you can see JAK3 and JAK2, so you can see that they're pretty highly active against both 3 and 2, and 1 should be on here somewhere, but you can see the numbers, and you can see where they're coming from. So bioorganic chem letters, you can see how much data is coming from the patents. There's a ton of data coming from the patents. The data against ABL was reported in a different paper, and you can see that there's no activity against ABL. And I'll note here that it's not just broadly under ABL, but you can see specific variants of ABL here. So here you can see actually 3 different variants of ABL and the activity against those 3 variants. So it's all the important information that you'd be looking for is captured by RMC. It's not just broadly like in Campbell. It would just be under ABL, and you wouldn't be getting all these nuances, which is very important information for you to be doing your research. And then it's going to show the next 200, and it's going to start going through all that data. So FIN and CDKL, etc., so on and so forth. So I'm going to go back to the query. I'm going to do one other type of search. So you can do a substructure search as well. So we can just click on these and get rid of this kind of this data in these fields, and I can just go and do a substructure search. So I may have an idea, let's say on a scaffold, and then I want to see all of the in vitro data on if this scaffold is actually known and what kind of data. So I actually had a scaffold I was working on 20 years ago, and I just want to see how much biological data was on this scaffold. So I'm going to choose this adamantane, and I'm going to do a little bit of modification on it. So I was working on a series of bispidone compounds, and so if you're not familiar with Reaxis at all, the drawing package is the Chemaxon package, and so if I could just spend maybe a minute on this, you can draw structures, or you can go to the templates and get templates, and then we can modify this. There's a number of different, I don't know why I did that. There's a number of different bond variations here, so you can see you could define any bond, single, double aromatics, single aromatic just a wide variety of different ways of doing a substructure search. You can go to the periodic table. We can create atom lists. So I could choose on a list of atoms and transfer them to my molecule. The basic atoms are over here. I can go to advanced query. I can use all my generic atoms, like any all metals, any halogens, heteroatoms, any atom except hydrogen and carbon, etc. I can create R groups as well. In this case, I'm going to look for I'm going to look for any, and then I'm going to look for any compound that has nitrogen at these two positions, and this can either be open, so I put the hydrogen there, or it can have a carbon, nitrogen, or oxygen at that position. And then I'm going to lock all the other positions down by adding the explicit hydrogens, and then I'm just going to remove the hydrogens from the two nitrogens. So I'm looking for compounds that have attachments at the two nitrogens here, and then there could be basically anything else at that position. And I'm going to leave the target open so I can find any targets that they were active against. And I'm just going to go under the bioassay category, and I'm only going to look for in vitro efficacy. And here again, it's going to bring me into a heat map. So it found actually 1,300 compounds. And here it's nice because you can see that they're active against a wide range of targets. Not every compound's active against every target, but I can scroll down through this, and I can actually organize these by safe factor 10A. I was interested in just seeing all the most active compounds against 10A. I could sort any one of these compounds or any one of these targets in a descending manner, and then I can just look for all the most active compounds against. So here there was only 8 or 9 compounds that were active. Then I can view the structural information for those. If I hover over it, it's going to show the structures. I can do that in like manner here for the nicotinic receptor. And here there's quite a number of compounds that were active. And so I can begin to scroll through these and get an idea of what types of compounds are there. So this can be important to me, understanding are these compounds known, what's their activity, can I take this data now and say go to the substance report. I can export this data as either an SD file or as an Excel spreadsheet. And if I export it as an Excel spreadsheet, I can limit the data to just the hit data. In this particular case, that was all the biological data. But I can go back and do another pointed query and just say I only want to look at all the nicotinic activity. Why don't we actually do that? Because that will make it more meaningful. So we're going to go up to target, and nicotinic acetylcholine would be actually we can just start typing it. There you go. nicotinic acetylcholine, and I'm going to just say receptor. I'm going to say all because I don't remember if it was a receptor or one of the other. Oh. There we go. And then we're going to search for that bioactivity again. And there's 271 compounds. So we're going to go to the substance report. We can look at the data, and here we could see this compound was marginally active, but it was against the nicotinic acid receptor. And then we can export that as an Excel spreadsheet, hit data only. So instead of it finding all the data, instead of it exporting all the data, it's only going to export the hit data. And it's also going to export the structure as an SD file into the Excel spreadsheet. And now you can take this data, you can do SAR and other tools, you can import it into a tool where you can do calculations on this. You'd be able to do, we do all the Lipinski calculations automatically, and I'll show you that in the Excel spreadsheet, but you can do further calculations on these compounds and decide which ones would, and then you can do a comparative analysis between activity and calculated properties. And as soon as this writes out, we'll look at this, and then I'm just going to open it up for questions and see if see what you guys think. And so here it's writing out the SMILES file as a structure, and then it's giving me all the identification information, the CAS numbers, the reaxis IDs, and then all of the calculator properties are here, the log P, the hydrogen bond donors and acceptors, the polar surface area data. There's a Verber count here as well. And then if we scroll over, it's going to show me the target, any ancillary information, target subunit names, and that's important as well. We can sort by the subunit if we want to eliminate some of those that we weren't interested in. And then we have all of the data here. And in this particular case, so you can see that it's writing out either KIs or Emaxes or EC50s, etc. So if we wanted to do a true comparative analysis, we may only want to look for those compounds that had a KI on them, and then we can just write out all the compounds that had a KI, and then we can do a comparative analysis. And then also all the references are under the reference field here. So you can click on that cell and it's going to show every single reference that's in that cell. So that's a quick tour of reaxis medicinal chemistry. I thought that we can open it up now for questions and see what people think, and if anybody has any searches that they want to do, and